Hi everyone, this is the day 28 mini lecture recording. There's no quizzes today. You can earn extra credit towards your daily points. The extra credit is due at the end of our normal class time on Wednesday, so December 6th at 3:15 p.m. There's an activity sheet and there is a Blackboard survey. So take a look at the day 28 activity sheet. It'll have all of the directions for earning those extra credit points. Please pay attention to all of the information for our final exam that's listed on this slide. Our final exam is Monday, December 11th. The study guide is extremely important. The review sessions are listed correctly there in 1B. Okay, so we'll have review sessions on Friday at 5, Sunday at 4. Please ignore the review sessions listed in blue for number 2. Those are incorrect. Okay, so today we have a fun topic, which is the real-world applications of genetics. So first I'm going to tell you a little bit about some genetically engineered organisms and edible vaccines. We'll talk briefly to reinforce the impact of the Human Genome Project on society, talk a little bit about transgenic animals, and end with CRISPR. So the first example of a real-world application of recombinant DNA technology is insulin. So this is recombinant insulin that's used by many diabetics. It was the very first human gene product made by this technology back in 1978. And this insulin is produced using bacteria. So this is a little bit of a refresher from Module 2, where we learned about cloning different DNA fragments into plasmids, which is shown at the top of this slide, where we see the two subunits of insulin, the gene A subunit on the left and gene B subunit on the right. So these were separately cloned into two plasmids. They were transformed into E. coli, the proteins made in the bacteria, and then towards the bottom of the screen, you can see that proteins are purified and then recombined to generate active insulin. There are now over 200 recombinant products that help our society in a variety of ways. Recombinant DNA technology is also useful in vaccine production. Here we can see a gene from a human pathogen is inserted into a vector or a plasmid. In this case, the plasmid is introduced into plant cells instead of bacteria. These leaf segments then sprout into whole plants, such as bananas, and then eating the banana will trigger an immune response to a pathogen. So this technology is extremely useful in developing countries where there's a lack of refrigeration, a lack of sterile conditions, and a fear of needles. So edible vaccines are definitely in lots of clinical trials using bananas and potatoes. Next, I want to come back to the Human Genome Project. We talked about this back on day 20, I believe, but today I want to just reinforce and emphasize the social impacts. So you know from both lecture and lab that the human genome sequences are over 99% identical between any two people. And you also know that there's more differences within geographically distinct groups than between them, which I think is really surprising to most people. We know there's a lot of benefits from having the human genome sequence, and these are listed here, and we talked about those. But today I want to emphasize some of the concerns and social impacts. This is really important for the final exam to understand these concerns. There is definitely a concern about how information from genetic tests will be used. Right, so it's great that we have new genetic testing and there's this increased technology allowing us to answer more questions than ever before. So some of these concerns are about how insurance companies should be allowed to use genetic information in making decisions about employment and insurance coverage. There are privacy issues. Who should have access to your data? Should your relatives? What about embryonic screening? Should information be allowed to select traits of offspring? And lastly, should companies be allowed to patent genes and to sort of profit from this kind of exploration? So many different concerns, and it's important for us to understand both the technology, but also these ethical concerns about how we use the technology. Okay, the next topic I want to talk about are transgenic animals. So we talked a little bit about how you can inject DNA into a newly fertilized embryo and then implant that injected egg into a female. So shown on the left is a needle microinjecting into a newly fertilized mouse embryo, and that would be similar to what we saw with the SRY experiment, right? When SRY was injected into an XX egg and then, right, the phenotype was examined. And so in this case, this is human growth hormone, DNA, that's being injected. And you can see in the phenotype on the right, we see a transgenic mouse that's now expressing, in this case, it was human growth hormone that was injected. And we can see that mouse with the arrow is much larger than the control mouse on the right. So what does this experiment tell us? First of all, it's a model for gigantism, which is a human syndrome that does occur. We could also potentially learn how to accelerate animal growth. But one important point for activity 2 is that neither the DNA sequences nor the amino acid sequences need to be exactly the same to either restore function or give function. In this example, we see a human gene for human growth hormone is able to function in the mouse. Now, although these are both mammals, you also know there's many differences between humans and mice. So the DNA sequences for this gene are not identical. And yet, right, this gene can be made into a protein that is functioning very similar in the mouse. Okay. Agricultural example of transgenic animals. Agriculture, right, is big business. When we look at cows and mammary glands that are susceptible to infection, this accounts for $2 billion in losses in the US dairy industry. And so one solution is to express the lysostaphin gene to kill Staph aureus before inflammation occurs. And you can see in the graphic there in the cartoon kind of how that might happen. There are other examples, potentially trying to generate transgenic cows that are lacking a prion gene to avoid mad cow disease, which has been a big concern over the past couple of decades. We can also make knockout transgenic animals. So shown here is a little cartoon about how one might do that. This is a little bit more complicated than just expressing a gene in a transgenic mouse. Here we're actually wanting to deplete the expression of a gene. And so there are a variety of different ways to do this. And one of them is shown here where embryonic stem cells are being used. So you're actually knocking out the gene in the embryonic stem cells. So these would be cells growing in a petri dish. And then you can take those cells and put them back into the mouse. So that's one way you can do that. What's really helpful about generating these knockout transgenic animals is you can model human diseases. For example, if a loss of a tumor suppressor gene, right, that can cause cancer. There are different disease models using knockout mice for obesity and heart disease, etc. So this is really helpful technology. And again, the key idea here is that neither the DNA nor amino acid sequences need to be identical. And so it's really important for the final exam to take a look at activity 2, apply this concept and what you learned previously about the genetic code and the degeneracy of the genetic code, as well as mutations and the idea of silent mutations in activity 2, right? So this is very exciting technology and will continue to be really helpful, I think, in the clinical setting. Okay. The last major point I think I want to end with here is CRISPR. So CRISPR technology, CRISPR is extremely important. It's been in the news a lot because it's so exciting. The Nobel Prize was awarded recently for this discovery. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. So you can see why we just abbreviate that to CRISPR. So what it is, it's a gene editing technique. And why it's so exciting is its specificity. It can target a very specific section of DNA, make a precise cut or break at that target site, and then it can either make that gene non-functional, or you can replace one version of a gene with another. Okay, so what are some potential applications of CRISPR to human health? One application is what we were just talking about with those transgenic knockout mice. So you can, instead of using the older technology that was a little clunky, you can generate mutations very specifically in genes if you want to explore the resulting phenotype or make these animal models that phenocopy human diseases. Even more exciting, I think, is that CRISPR can be used to correct a specific mutation or introduce a new sequence like a green fluorescent protein tag if you include something called a repair template DNA. And I've listed a couple of links if you're interested to see more details on how the technology works. In the Bio 545 class, Molecular Genetics, we talk a lot about CRISPR. One potential application of CRISPR is shown here. Researchers have used CRISPR to cure a type of liver disease in adult mice. This is type 1 tyrosinemia. It affects 1 in 100,000 humans. It occurs due to a mutation in the FAH gene, and it's very serious. It results in liver failure. So what researchers did is they injected the wild-type FAH CRISPR into veins of diseased mice, and in a very low percent of those liver cells, the mutant gene was replaced by the wild-type copy. So that sounds like really low efficiency, but those liver cells multiply very frequently. So those edited cells went through mitosis, right? They multiplied, resulting in about 1/3 of the liver cells with restored function. And this essentially was a real cure, not just a treatment, but a cure for these mice that had this disease. So this type of research is a really important step towards developing new gene therapies in humans. We might also be able to use CRISPR and gene editing as a solution to our shortage of organs for people who need organ donations. Pigs hold a lot of promise, but there's problems with immune rejection and viral infection. So what people did here is they did some research using CRISPR with pig embryos in culture to disable 62 viruses that are embedded in the pig genome, and then also separately altered 20 genes that cause immune responses. So the idea here is that you can use CRISPR to target and gene edit not just one gene at a time, but many different genes. And what they found was that the embryos looked pretty normal in culture, but this hasn't progressed to having living animals yet. There are ethical questions that kind of come along with this technology. And so in 2017, the US National Academy called for caution for any human germline gene editing, right? Okay, I mentioned the Nobel Prize was awarded recently for the discovery of CRISPR, and this was back in 2020. Emmanuelle Charpentier and Jennifer Doudna shared the award for developing this technology, and this was the Nobel Prize in Chemistry. Right before the Nobel Prize was awarded, there was a lot of news articles and headlines about CRISPR in the news, and this was actually kind of negative. So the first CRISPR-edited human babies were reported at a Hong Kong conference. Okay, this was before 2020. And this was by a researcher, Dr. He, and the CRISPR pioneer, Dr. Jennifer Doudna, was horrified, right? We just said that we don't maybe quite know enough. We're not ready for human germline gene editing. The NIH director at the time, Francis Collins, called it profoundly disturbing. And another prominent researcher called it monstrous. Okay, so why were people so upset? So even the scientists in Asia, in China, said this baby gene editing was crazy. Over 100 scientists condemned this report as crazy and unethical. His claim was that he had altered the genes of twin girls to create the first gene-edited babies. Okay, and so you can see some headlines here. This was back in 2018, right? And people are saying this is a step too far. Okay, so you can think for yourself what concerns you might have regarding using CRISPR to edit genes in babies. I only briefly mentioned this idea of off-target effects, but the idea is that sometimes when you are injecting the machinery, the CRISPR machinery, with that precision, that it's not perhaps 100% precise. So if it's going to affect other genes than the ones you intend, there could be problems. Okay, so more information about what was happening here. His specific gene editing experiment with these twin girls was to change the CCR5 gene, which is used by HIV to enter cells. And individuals with a naturally occurring deletion mutation, it's called Delta 32, are resistant to HIV. You may have heard of these individuals. Okay, so we need to understand that it's a very small number of individuals that seem to be resistant to acquiring HIV, even after repeated exposure, right? So back in 1996 was when we realized that those resistant individuals all had the exact same 32 base pair deletion in the CCR5 gene. And here's a little picture about the CCR5. It's shown there in pink, and it acts as this receptor that is needed by HIV to enter T cells. Okay, so what Dr. He focused on was changing the CCR5 gene to generate this deletion mutation. Okay, so basically his goal was for these twin girls to be resistant to HIV. But we do need to realize that neither infant had HIV, nor did the mother. Okay, so how does that information affect your thoughts about concerns people might have about what he did here? You can think about that for a minute. So Dr. Hay described his team's research at a conference, but it had not yet been peer-reviewed or published. And based on the slides he presented at this conference, some scientists had concerns that the work was not even executed well. The results indicated actually that neither girl actually had the correct mutation, this Delta 32 mutation. Lulu, one of the twin girls, had a different mutation in CCR5, and the second twin girl, Nana, had 2 different mutations. Okay, so that is definitely a concern that he thought he was designing this CRISPR experiment to achieve one specific kind of mutation, but it didn't seem to work as he intended. So as you reflect about this new information, you can continue to think about your concerns. If we were in a group setting, we would have, you jot down some thoughts and then have a group discussion. But obviously we're not doing that today. So based on prior classes, I would guess that if we were in class discussing this together, some of the concerns we would have would be that we can't assume that these other mutations in the general area of Delta 32 are going to be beneficial. 2 of the 3 mutations in these twin girls are pretty substantial changes in the CCR5 gene. It's never been tested before in a model system before trying it in humans. So it really hasn't been vetted. And this researcher was showing a real disregard for sort of the rules and conventions by which researchers do a step-by-step process before applying this technology to humans. Dr. Hay told the family that participated in the trial he was working on an AIDS vaccine development project. The informed consent document he gave the family used very technical language. And a news article was printed saying the father of the twin girls had only a high school background in biology, that the family was not told about off-target risks, nor were they told that gene editing was controversial and illegal in humans in 2018. So as you think about this part of the story, how does that affect any thoughts you might have about concerns and informed consent and about what this researcher did? You can think about that. As we kind of delve into even more specifics about what was happening here, the actual consent form was not even a real consent form. It was missing really key information about risks, and it was more of a business form, absolving the researcher of legal responsibility and giving his team rights to pictures of the babies from magazines and billboards and marketing. I mean, this is really pretty terrible. More information about this, he was acting under a cloak of secrecy, which means he didn't inform his institution, the Southern University of Science and Technology. He took unpaid leave to do these experiments in secret. And the claims that he made that he received ethical approval from this particular hospital were not held up. The hospital says there were no meetings. Okay. So as you think about his actions, this might affect your thoughts and kind of about the seriousness about your concerns. So the university conducted an investigation into the project. They called it a serious violation of academic ethics and standards. The signatures on that ethical approval form may have been forged. So the researcher's lab webpage was taken down. And what's really kind of amazing in this story is that the researcher, Dr. Hay, published a separate paper on ethical principles that he himself then violated. So there's some real kind of contradictions occurring here. If you're interested, there are some really interesting films about CRISPR, some documentaries, so I'm showing you those here. There was a 4-part miniseries on Netflix back in 2019, and then another one called Human Nature. So these documentaries cover some of the really exciting potential of discovery as well as these ethical concerns. Okay, so we kind of fast forward in our story back to Dr. Hay. So there he is. And you can see here this news caption that he shocked the world by declaring he had successfully altered the genetic code of 2 babies. He was met with overwhelming skepticism and condemnation. And then the case gets even weirder. The South China Morning Post reported the infamous scientist went missing. And he was missing present. Okay. And now, fast forward to June of 2023. So he got out of prison after about a year, and now he is back in the lab again. And I put the link to an NPR article that was published recently. He claims that lots of individuals and family members that are affected with Duchenne muscular dystrophy are asking him to help, basically. And it's really unclear, I think, what this means for the scientific community. He's under a lot of restrictions, and he, is not allowed to do a lot of different types of research at this point. But the fact that he's back in the lab at all definitely upsets a lot of scientists, I would say. Okay, so now switching over to kind of more exciting positive news. Just a few weeks ago, November 21st, 2023, the world's first CRISPR therapy is approved, is the headline. Who will receive it? And so this is a company called Vertex, and they have gone through the correct process, stepwise process, of getting gene editing therapy for sickle cell disease and beta thalassemia approved. Okay, so this is extremely exciting. These are really serious diseases for those who suffer from them. We have not had good treatments for these diseases to date. So, and certainly we have not had cures. And this is the idea that this could actually be, cures. So now the new challenge is the cost of this treatment. So I've put the link there if you're interested to read more about it. This is extremely exciting, though, and I think is an example of using this technology the right way, with the right purposes, and it really is very exciting, and I think it is already revolutionizing medicine and will continue to do so. Okay, so as we wrap up, I just wanted to share what the Bio211 team hopes you learned this semester. You can see we have this sort of green triangle where we have our first 100 level courses, Bio 111, Bio 112, building this base. Our course, Bio 211, hopes to build on that foundation, along with Bio 213 ecology. And if you continue on with a biology major, you'll go on to take Bio 212, form and function, and Bio 500, biostatistics, and you'll pick electives. And we hope all of this really is preparing you well for a career. In our courses, we want to emphasize discussions and applications and problem solving. We hope to make some connections between lecture and lab, and you can see all of the core skills listed there in pink, as well as core concepts. And in our course, Bio 211, we've definitely talked about evolution and evolutionary conservation. We've talked about structure and function just a little bit, and definitely a lot about information flow. That's the central dogma, and how is that information, genetic information, stored? That's in DNA, the genetic material for nearly all organisms except RNA and some viruses. Okay, so we hope you've learned a lot. We hope you're able to take some of what you've learned with you as you go into future courses and certainly in your lives. I want to thank you for a great semester. It's really been fun working with all of you this semester. I hope you keep in touch. Don't forget to think about summer opportunities. Maybe once finals are over. These internships for summer are posted now. I'm going to post some more links in Blackboard soon. And again, if you're interested in learning more genetics, I've listed a couple of the courses here.